Have you ever thought about what it would be like to give a living thing a superpower? Well, today, we're going to dive into the amazing world of genetic engineering, where scientists are doing pretty much that with some of the simplest organisms on our planet. Let's see how it all works. I mean, it sounds like something straight out of a comic book, right? But this is a real central question in modern biology. How on earth do you take a single-celled organism, a little bacterium, and totally change what it can do? And we're not just talking hypothetically here. We're talking about giving these bacteria real, observable new abilities. Things like, say, the power to survive in an environment that would normally kill it, or even the ability to change its color right before our eyes. So how is it even possible? Okay, so to pull off a mission like this, you need the right toolkit. And in our case, that means we're going to need three key things: a test subject, a new set of genetic instructions, and a very specific environment to make the magic happen. First up, let's meet our volunteer. This is E. coli. You've probably heard of it. It's one of the most well-studied organisms in all of science, which honestly makes it the perfect candidate for a genetic upgrade. Think of it as a reliable blank slate for our experiment. And if E. coli is our computer, this is the software we're going to install. It's called a plasmid. It's just a small circular piece of DNA. The easiest way to think about it is like a tiny biological USB drive. And this map you're seeing shows the specific programs, the genes, that we're going to load into our E. coli. So this particular plasmid, called PGAL, is carrying two really important genes. First, there's the AMPR gene. This one's a lifesaver. It acts like a shield, giving the bacteria resistance to an antibiotic called ampicillin. The second gene is for an enzyme called beta-galactosidase, and this one is our signal flare. When this enzyme meets a certain chemical, it triggers a reaction that produces this awesome blue color. So you've got one gene for survival and one gene to show us that it actually worked. All right, so we have our bacteria and we have our genetic software. Now comes the really tricky part. How do we get that little USB drive, that plasmid, into the E. coli?
The official name for this whole process is bacterial transformation. And believe it or not, it's something that can happen in nature. Scientists have just figured out how to harness it in the lab. And mastering this isn't just a cool party trick. It's the fundamental skill behind making bacteria produce all sorts of incredible things for us.
This flowchart right here is our whole plan of attack. Now, look closely. You'll see there are two paths. Over on the right, that's our experiment group. They get the plasmid DNA. But on the left, we have a control group that gets everything except the DNA. And this is absolutely crucial. By comparing the two at the end, we can be 100% sure that any changes we see are because of that plasmid and nothing else. Now, you can't just, stir the plasmids into the tube and hope for the best. You have to convince the E. coli cells to actually take them in. The technical term is making them competent, which basically means making their cell walls just porous enough to let that new DNA sneak inside. And the way we do it, well, it's surprisingly dramatic. It's a technique called heat shock. It's wild. First, you chill the cells way down on ice. Then, bam, you plunge them into a hot water bath at exactly 42 degrees Celsius for just 90 seconds. And then, just as quickly, it's right back onto the ice. This crazy temperature roller coaster puts stress on the cell membrane, creating these tiny, temporary openings, just long enough for those plasmids to slip right inside. Okay, the procedure's done, the cells have had a little time to recover, and now it is the moment of truth. How in the world do we know if any of our E. coli actually accepted their new superpowers? To find our successful recruits, we spread all the bacteria out onto a petri dish. But this isn't just any old petri dish. This dish is actually a test. It's loaded with that antibiotic, ampicillin, which is totally lethal to normal E. coli. And it also has that X-gal chemical, the one that will turn blue if our special enzyme is there. And this, right here, is where it all comes together. It's so cool. Look at the control plate, the one with the bacteria that got no plasmid. Nothing. It's completely empty. The antibiotic did its job and wiped them out, just like we expected. But on the experiment plate, we see colonies. These are the descendants of the few bacteria that successfully took up the plasmid. Their new AMPR gene, that shield, protected them and allowed them to grow. And because they also have that second gene, they turn the X-gal blue, basically shouting to the world, hey, we've got superpowers. Now, in science, just saying it worked isn't quite enough. We need to know how well it worked. And that's where a measurement called transformation efficiency comes in. It's a specific calculation that moves beyond a simple yes or no and tells us exactly how many of our bacteria were successfully transformed. So, for this particular experiment, the result was about 6,400 successful transformations for every single microgram of DNA we used. That might not sound like a huge number, but in the microscopic world, that is more than enough to start a whole thriving population of super-powered bacteria. This kind of precise data is what lets scientists compare different experiments and keep getting better at what they do. Okay, so making bacteria turn blue is a fantastic proof of concept. It's a great lab experiment. But why does this one simple idea matter so much in the real world? Well, let's zoom out from the petri dish for a second and look at the massive world-changing impact of this technology. This exact process, this idea of inserting a piece of DNA into bacteria to make them produce something new, is the engine behind countless modern miracles. It's how we turn bacteria into tiny factories that pump out insulin for people with diabetes. It's how we engineer crops to fight off pests, how we develop brand new vaccines, and even how we create the enzymes that make our laundry detergents work better. The principle is exactly the same as making that blue colony. So what does this all mean? It means we've learned how to literally rewrite the code of life on a microscopic scale. What started with a simple experiment, a few blue colonies on a plate, has unlocked a world of possibilities. Which really brings us to the biggest question of all. Now that we have this incredible power, what problems should we ask these tiny organisms to solve for us next?